So I think the slides are visible and I am clearly audible. So welcome to the week 8 section of the course Cell Culture Technologies, organized by NPTEL, that is conducted by for the teaching of TAship, conducted by IIT Madras. So I'll just move on to the first question itself for the week 8 section. So for the question number 1, which of these can be used to separate population of two cell type from a mix? First one immunopanic, second one fluorescent assisted cell sorting or FACS, both A and B, or none of the above, which might be the answer. Okay, I am getting option number both A and B. Option C is yeah. So the answer to the question is you are right. Option both A and B, both immunopanning as well as FACS, fluorescent assisted cell sorting. So if I give you a brief explanation of what the techniques are, I will just start with immunopanning. So both are used for basically separation of cell from a population of cells, right? Different type of cell population that is present within a sample that you are getting. So if you are like one of the common techniques that we earlier discussed in the last week is density gradient centrifugation, right? So in that case we are taking a tube there and then we are adding a different gradient of sucrose or any other solvent mixture you are using, or OptiPrep or anything, starting from the lower concentration to the higher concentration. Then you are doing then you are putting the sample at the top, higher concentration to lower concentration, then you are putting a sample at the top, then you are undergoing ultracentrifugation there. So based on the density of the particles that are present within a sample, the the different molecules or different composition that make up the sample gets differentiated based on the density that they are having, based on the density as well as size as well as the shape. So based on that the sample get separated, right, in case of density gradient centrifugation. So what if we have two different components within that particular sample having the same density? In that case the sample having the same density appear at the same layer, right? So we might be thinking that it might be the same population, but in this case if you look into the molecular composition and all, they might be different, or it might be different cell population there. So in that case we will apply like a more complex technique like an immunopanning. So it's similar to ELISA. So basically what we have is like a particular sample. This is your sample basically, you can see that here. And then you have a Petri plate that is coated with a particular antibody there, or we first incubate the sample with a particular antibody. Suppose that this particular antibody binds to a particular collection of cells there basically in this case. So if you see that there are different colored cells, right? So this primary antibody actually binds to this particular brown colored component that is present in the sample. Then you are transferring that that antigen. In this case we will consider this as an antigen and this as an antibody there. So if you transfer this particular incubated sample into a Petri plate containing a secondary antibody that actually binds to the primary antibody and incubate it for some time. So what happens is that this primary antibody, the Fc region of primary antibody interacts with this particular region of secondary antibody, and then they form a antigen primary antibody secondary antibody complex there. So all the other components that do not interact with the primary antibody is left suspended within that particular solution. Then you are giving a particular wash there. So omit this particular like step. It actually I think it's different there. So then we are washing the samples there. So all the components that are present in the sample that is unbound to the primary antibody is actually getting washed out. So basically we are using a buffer containing a particular detergent or any buffer, normal buffer there, to wash out this suspended sample, or just you are aspirating it out, and for the removing the remaining sample that are present within the lowermost portion of the Petri dish, you are actually using a buffer just to wash it out. So in this case you can see that the sample that has actually interacted within the antibody is actually left within the Petri dish, and then it is eluted out. This sample is eluted out into a new sample. So if you compare into a new tube there, so if you compare from the final tubes, the final tube with the first tube, you can see that there is a mix of population there. In the later on in the final tube, you can see that only the particular cell population or a particular antigen is there that shows interaction with that particular antibody itself. So this is called as immunopanning. Using a particular antigen-antibody interaction, you are usually separating a particular cell population from a particular cell from a population of different kinds of cell, so that you can actually do some cell culturing and all. So this particular technique is called as immunopanning. Coming on to the next one, it's also called as fluorescent assisted cell sorting there basically. So basically from the name itself, we are using a fluorescence, a laser, to actually sort out the cell, to analyze the cell population. From that particular population that you are getting, you are actually separating out different populations of cell into different sample vials that you are collecting. So first you have to label the cell using fluorescent dye, and that particular fluorescent dye might be attached to an antibody. So basically it's a fluorophore that is attached to an antibody, and this marks out certain target cell. So if you are marking out certain target cell, there might be particular receptors that are innate to that cell that might be expressing at the cell surface. So this particular antibody that we might be using have to target these particular receptors. In case of immunopanning also, it is the same reason. You have to have an antibody that targets a particular cell surface receptor that are present within the cell surface there. So if you have an antibody that you can conjugate it with a fluorophore, and then you can introduce into a population of cell. So what this does is that it actually binds to the particular cell surface receptor that is present on that single population of cell itself, rather than the mixed population, all the population. So after that, you can see that this is the mixture of cell that has been collected. So you actually pass the cell through a small you put the cell into a chamber, then it actually passes through a small capillary tube. So the size of the capillary tube is in such a way that it only allows a single cell to pass through it each time. So you can see from the figure that only one cell is passing through it, right? So the size is actually recommended to maintain in such a way that only a single cell can pass through that particular tube there. So when it passes through a that particular tube, there is actually a sensor that is present here. So basically the sensor is actually a laser that actually detects this particular cell population. So from the laser actually illuminates that cell, and based on the scattering, the size of the cell, scattering and the size of the cell, or the dense scattering that is produced from the cell, it emits a reflected light. So from that reflected light is then collected by photomultiplier tube, which is a detector which passes on the signal to a computer which analyzes the data. So based on the cell population, whether the cell is dense within, where the cell is not dense within, or whether the cell is size of the cell is different from one of the population that just passed through, you can actually compare the cell diversity that is present within the sample, and then it passes on through a particular end of the tube, which actually mixes with a particular liquid, which gives it a drop-like appearance. On that basis, then it is then given an innate charge based on an electromagnetic field that is passing through this particular region. So based on the presence of the electromagnetic field and this droplet that is present, the cell actually has a tendency to get ionized in this case. And also on the basis of the fluorophore that is being attached to the particular cell, the ionization might be different. It can be positive or negative, or it might have no charge at all. On that basis, after all, when it passes through the electromagnetic field, then it has a tendency to get separated based on the innate charge that it just got when it formed the droplet there. So you can see that the negatively charged actually moved to the positive side of the the electrode there, while in case of the no charge at all, it moves onto a center, it just drops down, while in case of those cells having the positive charge, it moves onto a next collection vial there. So basically on this basis, the cells are actually getting separated from a population of cells there, and also you get an idea of how dense inside the cell is, whether if you have a cell population, like if you are studying a blood cell population, right, leukocytes are there, macrophages are there, some of them have particles within it which gives it a much denser appearance, some of them might not have. So through this technique you can also study the cell and sort out those cells, so that you can do a cell culture if you want a particular leukocyte, or if you want to grow a macrophage there. So That's the basic principle of a fluorescent assisted cell sorting there, and we have also discussed the immunopanning. So does anyone have any doubt regarding this question? Should I explain it once more? Okay, so okay, I will just repeat now. So this is the immunopanning. Actually, there is some mistake here. So this should be the brown particle that should be present here. So I haven't also observed that. So yeah, so this is the immunopanning. So yeah, so as far as I read, when this is this made into a droplet, right, it's actually getting ionized. So I also need to know how, like also need to read how it gets ionized there. Then it passes through this electromagnetic field and then gets separated. So that's also a particular, like I also need to search that out. I think it's when you are getting an what you say mass spec also, when we are making a spray of cells or something, right, sometimes it gets ionized in this case. So that ionization process is also there. Okay, so I think this is clear. So I will just move on to the next question. So coming on to question number 2. Which of these is a signature for all leukocyte group? CD45 plus, CD45 minus, CD340 plus or CD340 minus? Okay, I am getting option number 1. Any other option? Okay, so basically you are right. The answer to this question is CD45 plus. So basically these are cell differentiation marker, right? CD stands for cell differentiation, cholesterol differentiation or cell differentiation. I also don't recall that properly. So basically this all the leukocyte groups have this particular cell surface marker or receptor, that is CD45 plus, and it's a common leukocyte antigen there, that is expressed in all hematopoietic or blood cells there. And it is used for several immune function, and in this case in case of leukocytes, basically their function is immune response, right, regulation of immune response. So this is CD45 plus means that the leukocytes having this particular cell surface marker. If a particular leukocyte lack this cell surface marker, then it's called as CD45 minus. However, if that particular leukocyte does not have the cell marker, then that is not a characteristic of that particular leukocyte, right? CD340 plus or HER2 marker is actually associated with certain epithelial cells and some tumors. So basically in tumor, and it is also a differentiation case, but if you want to grow any epithelial cell or tumor, you can usually use CD340 marker, but in case of leukocytes and all, as CD45 plus marker is a common leukocyte, you can actually use that particular marker there, right? So CD340 minus meaning that that particular marker is absent there. So basically if you want to do immunopanning of any leukocyte or if you want to do a fluorescent assisted cell sorting, that is FACS in this case and all, you can basically use this particular marker as a target for several antibodies there, right? Because it is actually important to this particular leukocytes itself. So basically you can introduce a antibody that targets this marker and then do a fluorescent FLAX or a immunopanning, whatever is necessary to separate them from a population of cells there. So I think this is quite clear. So does anyone have any doubt? Okay, HER2, I need to check that out basically. HER2, I don't know the particular answer to that question. Okay, so I will discuss it within the next week if I get the time. So right now some of them I also have doubt. So yeah. So does anyone have any other doubt? Okay, so I think it's quite clear. So I will just move on to the next question. Question number 3. So, and dash is a protein molecule that recognizes dash on a cell surface, antigen, antibody, antibody, antigen, 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 or antibody, antibody, which might be answer. Yeah, I'm getting option number 2, antibody and antigen. Any other option? Okay, basically you are right. So the answer to the question is antibody and antigen. So an antibody is a protein molecule that recognizes the antigen on the cell surface, right? So basically, if I give you a diagram, basically this will be the one. So this is an antibody, right? So there are heavy chains there, there are light chain there. Basically, it is a dimer, right? So you can see the variable region that are present at the surface, right? So the antibody has a particular variable region that promotes a binding to a particular substance called as antigen there. So antigen also should have the ability to bind to this particular region that is present at the surface of antibody there. Only then a particular interaction called as antigen antibody interaction can happen. So once this antigen antibody interaction can happen, several leukocytes or any other immune cells actually recognizes this bound antibody there, and then it elicits an immune response, a primary response or a secondary response, whatever is necessary. So this antigen has to particularly interact with this particular antibody to form that particular interaction there, right? So in this case you can see there are several substances. If you look into a particular human physiology and all, there are antibody that is present within our immunoglobulins that are present within our bloodstream, right? So whenever a particular foreign substance enter into our body and this interacts with an antibody, then it elicits an immune response there. So this particular foreign substance that is entering our body is called as an antigen. So it can give out a normal immune response, or it can give out an exaggerated immune response for what we call as something called as an allergy, right? So basically it is an allergen also in this case. So basically this particular function there. So even in the case of immunopanning we have discussed, right? So several of the components within the cell sample actually binds to the antibody, right? So that particular cell can be actually called as an antigen there, or a particular receptor that is present at the cell surface of that particular cell that is binding to the antibody can also be called as an antigen there. So basically giving rise to what is called as an antigen antibody response, which then helps us to separate out that particular cell type from a population of cells there. So to this question, an antibody is a protein molecule that recognizes the antigen on the cell surface. So do you have any doubt or or should I explain it once more? It's a basic question, basically. In case of undergraduation or post-graduation you might have learned it. So I think I will just move on to the next question. So coming on to question number 4, in where there are two types of motor neurons, one are small in size called what, and other large in size called what. First option is dorsal horn alpha gamma, second option is ventral horn alpha gamma, third option is dorsal horn gamma alpha, or fourth one ventral horn gamma alpha, which might be the answer. Okay, I am getting option number 4. Any other option? Okay, so basically you are right. So the answer to this question is, in ventral horn there are two types of motor neurons. One are small in size called gamma, and other are large in size called as alpha motor neurons. So basically these are the two different motor neurons. Basically motor neurons are something that is present within the ventral horn, right? So basically those are mostly sensory in nature. So if you look into the particular structure, this is a spinal cord there, that is a peripheral nervous system, right? PNS as well as central nervous system is there, and spinal cord actually forms a part of this PNS or peripheral nervous system. So this is the region where ventral horn is present. So in this region you can see that several neurons are present there, right? So this can be either dorsal or as well as These can be either alpha neurons, which are bigger in size, and other one is the gamma neurons, which are usually smaller in size. So basically, in case of a fetus, if you want to collect the motor neurons at the E14 embryonic day, you are basically removing the dorsal horn or the spinal cord from the particular embryo. Then you are removing out all the cell population that is present within that particular this ventral horn there. So what is destined to become a ventral horn there? So basically it consists of these particular neurons, that is alpha as well as the gamma neurons. So if you look into the functionality of a gamma neuron, basically this is the spinal cord there, you can see that this is the ventral horn. So several neurons are arising from here, right? So one is larger in size, while other one is smaller in size. So the smaller ones are called the gamma neurons. So this gamma neuron actually interacts with the intrafusal muscle fibers that form a part of the muscle spindle there. So muscle spindle is part of the our skeletal muscles that actually reacts to sensation that is getting from the surrounding environment. Then based on that, it actually contract as well as relax. So actually this function is under the control of what is called as a muscle spindle there, and this whole structure is a muscle spindle. So this whole muscle spindle actually on the basis of this particular motor neuron there, that is the gamma neurons that are interacting with the intrafusal muscle fibers, as well as the alpha neurons that are interacting within these extrafusal muscle cells there. So these two motor neurons that are interacting within the spinal cord with the muscle spindle actually tells whether the muscle has to contract or relax. So this is actually the function of this particular ventral horn there, and the ventral horn, once it receives the signal, it actually transmits to a brain transmits the signal back to a ventral horn, it then transmits the signal back to a muscle spindle there, telling it to whether to contract or to relax. So this is basically the function of the both neurons there, alpha as well as gamma motor neurons. So coming on to this answer, in case of ventral horn, there are two types of motor neuron. One are small in size, which is the gamma motor neuron, and the other one is larger in size, which is called as the alpha motor neuron. So does anyone have any doubt regarding this? Should I explain it once more? So I think this is quite clear. We have discussed in the last class also, so I will just move on to the next question there. Coming on to question number 5. In rat spinal cord at E40 only, motor neurons express a particular marker. This information can be used to isolate this cell from a mixed culture with other cells of the spinal cord, test electrical activity of this cell, digest these cells, none of the above. Okay, I am getting option number 1. Any other option there? Okay, so the answer to the question is you are right. Isolate these cells from the mixed culture with the other cells of the spinal cord, right? So basically, you know that if a cell has a particular like cell surface receptor or a particular marker present at the cell surface, then that particular marker can be utilized to separate it out using FACS or any other this immunopanning technique, right? So basically, some of the common markers that are present at that particular time range is chondrolectin, calcitonin related polypeptide alpha, or estrogen related receptor beta. So these are just examples, so I thought to give some wide idea. Might be that it is not important. So these are some of the receptors that are present at that particular time point of the rat spinal cord that can be utilized for cell separation and all. So I think this is quite clear. So does anyone has any doubt regarding this? Okay, so I think it's quite clear. So I will just move on to the next question. Coming on to question number 6. Trypsin is used to what? By digesting the extracellular matrix, as it is a lift the cell culture or the cultured cells of the surface. So it is a condensation enzyme. Lift the cultured cells of a surface, proteolytic enzyme. Attach the cultured cell of the surface, condensation enzyme. Attach the cultured cells of the surface, proteolytic enzyme, which may be the option. Okay, I am getting option number 2. Any other option? Yeah, basically the answer is right. So it actually lifts the cultured cells of the surface. It is a proteolytic enzyme. So basically not only cultured cells, but also when you are about to do a cell culture, right, you need that individual cells that are devoid of extracellular matrix and all. So in that case trypsin is also used, but trypsin as compared to other proteolytic enzymes like papain or collagenase etc. is much harsher in nature. So after incubating it for some time, you need to add some trypsin inhibitor to inhibit the activity of trypsin, so that that particular function is stopped there. So the trypsin won't only have removed the or degraded out the or lysed out the extracellular matrix without harming the cell as a whole, because we need the intact cell for the cell culture, right? So even after cell culturing, in order to lift the cultured cell out of the surface, which is bound to the extracellular matrix, you need to add some trypsin to do that basically. So afterwards also, once the incubation is done or the for some time, you need to add some trypsin inhibitor and just to stop the reaction there, so that the innate cell won't get degraded out. So basically it's a proteolytic enzyme, meaning that it degrades out proteins there. It degrades out proteins into smaller peptides there. So basically trypsin, if you consider, is a serine protease. So basically if you look into our body, basically it is secreted out by this pancreas, right? So basically the pancreas secretes the inactive precursor of this trypsin called as trypsinogen into the small intestine. So once it is released, when food comes into the small intestine, it starts to act on the food and it digests out the proteins that are present in the food there. So that's why it gave the name proteolytic enzyme, because it degrades out degrades out proteins into smaller peptides there. So basically it's a serine protease because of the presence of any serine residue that is present at the active site. So that is a common factor that gave it name, that is a serine protease there. And it also consists of other catalytic triad. It consists of other amino acids except from that of serine, like histidine and aspartate, giving it the name catalytic triad, because these two amino acids along with the serine makes the active site active. That's why it is called as a catalytic triad there. So it cleaves the peptide bonds at the carboxyl side of basic amino acids like lysine and arginine, and which are positively charged, and because of this the protein gets degraded out into smaller peptides also, from larger protein fragment into smaller peptide also. So that's why it's called as a serine protease, and it acts as a proteolytic enzyme. And because of this particular factor, because it can act with certain extracellular matrix proteins there, it is used as a degrading agent for this extracellular matrix, so that it can lift off the cell culture that is attached to a extracellular matrix proteins there, making it free there. So if you want to do any passaging, you can do that, or afterwards whatever you want to do. If you want to do a phenotypic reaction or any experiment, you can do that afterwards. So that's the basic role of trypsin there. So anyone has any doubt, or should I explain it once more? So I think it's quite clear, so I will just move on to the next question then. Coming on to the next question, question number 7. Which of these is not a component of B27 supplement? Vitamin A, biotin, acutase, or sodium selenite? It might be the answer. I am getting the answer. Acutase, option number 3. Any other answer? Okay, so basically you are right. The answer is acutase. So basically acutase is a natural enzyme mixture that exhibits both proteolytic as well as collagenase activity. So it is similar to a trypsin, collagenase or papain that is used to culture out individual cell from a from the like tissue cell, or if you want to remove a particular cultured cell from a what is a extracellular matrix or a culture plate, then you can actually use this acutase there. So for that, so it's basically exhibit both proteolytic as well as collagen-lytic activity, and the harshness is much less, basically is much more less active as compared to both these enzymes that we have discussed before. So basically it is not a component of B21 supplement, because media is something that should help the cell to proliferate and differentiate, right? So if we have something like a what you say a protein digestion enzyme or something like that, or proteolytic enzyme, it would basically prevent that from happening, right? So basically acutase is basically not a component of B27 supplement. And so, and also if you look into a sodium selenite, basically sodium selenite consists of this selenium, right? Selenium is actually a what you say basically it's an element, right? So basically it's a component of this particular enzyme, glutathione peroxidase. So glutathione peroxidase requires this selenium to be active. So when it binds to this selenium within the cysteine residue that is present within the glutathione peroxidase, it actually forms what is called as a selenocysteine there. So it makes the particular enzyme active. So this is actually peroxidase. From the name itself we can know that it actually prevents the oxidative stress from happening there, right? So hydrogen peroxides and other peroxides is something that is present or something that will form within some point when you are incubating a cell in a cell culture in an incubator, right? When it starts to growing, it starts to form all the free radicals and everything. So basically in order to prevent that, it is actually important for the activation of this glutathione peroxidase, which prevents the formation of this free radicals there, and also prevents it from oxidative damage there, which in turn prevents cellular injury. So it is something that is present within the vitamin B28 supplement there. So basically if you look into vitamin B27, you can know that it is actually a culturing media that is required for growing what is a fetal nervous tissues, right? So basically it consists of all the substance, this particular what you call vitamins, biotin. These are required for the maintaining the growth of the tissues, right? Sorry, the cells, right? So only then the cells will start to grow and proliferate. And B27 is actually used for growing this fetal nervous tissue, while in case of B27A, which is another subcomponent or a much more additional component of a B27 supplement, is what is required for growing the adult nervous tissues there. So B27 is used for growing the fetal nervous tissues there, while B27A is used for growing adult nervous tissues there. So osmolality also differs in this case. For B27 it is something around 220 or 210 milliosmoles, while in case of B27A is much higher because of the large amount of sodium chloride present there. It comes up to around 250 to 270 milliosmoles as far as I can recall. So this is basically a culture media. So these require these vitamins and also sodium selenite for activity in the presence of glutathione peroxidase, so that it can prevent the free radical accumulation there. Acutase, it is not needed because acutase is an enzyme that is a proteolytic in nature. So it will arrest the cell growth and differentiation there. So we probably don't want that in a cell culture or a cell culture system there. So the answer to this question is acutase. So does anyone have any doubt, or should I repeat it? Okay, so I think it's quite clear. So I will just move on to the next question. Coming on to question number 8. Glutamax is a dash, it's a dipeptide we know, and has what than glutamine? Which amino acid then? And so what is the answer? So I am getting the option number 2, L-alanine, L-glutamine, and also higher stability. Yes, basically you are right. So the answer to this question is Glutamax is a dipeptide, made up of L-alanine that is L-glutamine that is conjugated with L-alanine by a dipeptide bond, peptide bond, and has a higher stability than glutamine itself. So basically L-glutamine is an amino acid, right? So basically it is required in mammalian cell culture for promoting the cell growth as well as proliferation there, cell division, proliferation as well as differentiation there. So basically it also acts as an energy source similar to that of glucose, which acts as a ATP source there, right? It passes on to glycolysis and everything, then it starts to produce this ATP and all, so it is similar to that. So L-glutamine is important for the production of pyrimidine and pyrimidine nucleotides, which are basically required for the formation of this DNA, RNA and everything, right? Basically the purine and pyrimidine are building blocks of DNA or RNA or genetic source, whatever it is, amino sugars, glutathione, L-glutamate, and other amino acids, and are also involved in protein synthesis as well as glucose production there. So in case of aqueous solutions such as cell culture media, the L-glutamine has a short half-life there because it actually tends to degrade out spontaneously. So basically when it gets degraded out, whenever an amino acid gets degraded out, it actually starts to produce this ammonia and other carboxylic acid byproducts and all, which are basically toxic in nature. So when glutamine starts to degrade out, gradually the ammonia gets accumulated, and this can harm the cell culture and can in fact affect the growth of, affect the whole cell culture is there, leading to cell death and all. So what is done is that, so in case of long-term cell culture incubation and all, we cannot use glutamine, innate glutamine as a energy source there. So what is done is that a glutamax, a supplement is provided, which is actually a dipeptide, where this L-glutamine is actually linked to L-alanine by a peptide bond, and it is more stable in aqueous solution and does not spontaneously degrade. So whenever this cell wants to use this, or whenever the cell produces these aminopeptidases, it actually cleaves this or hydrolyzes this dipeptide, slowly releasing. It is not a fast release, it slowly releases this L-alanine and L-glutamine into culture media. So in this case, it is not only getting L-glutamine, it is also getting another amino acid L-alanine there, and this is a slow release there. This is a gradual slow release there. So this is why glutamax is important, because in many cases glutamine cannot be used as an individual amino acid source there. Instead, glutamax, which is a dipeptide, consists of L-glutamine and L-alanine, is used there, and because of the higher stability and the slow release in the cell culture media, it is preferred as a supplement or a cell growth supplement in case of culture media and all. So I hope this explanation is clear. So does anyone have any doubt? Or should I explain it once more? I think we have discussed it in the last 2 weeks also, so I think it's quite clear. So I will just move on to the question number 9. So coming on to question number 9. What is a supplement used for serum-free fetal hippocampal neuron culture media? B27, G5, collagen or fibroid? Okay, I am getting option number 1, B27. Any other option? So basically you are right, the answer is B27. So basically in case of serum free, serum means like it's actually a B27 is actually a defined media. It's not like a rich media as compared to wherever a serum is present there. It's not similar to L15 media or anything. It's not similar to what you say DMAM media. What is Dalbecos modified Eagles media, right? So it is not similar to that. This is a defined media, meaning that this particular B27 only have the components that is added based on our behalf. So if you are adding 1 gram of this substance, 1 milligram or 1 gram of this particular substance, we know that what is a particular substance and how much that substance is present within that particular media. So that is called, why it is called as a defined media there. If you are adding serum-free media, it becomes a non-defined media or undefined media, or what we call as a rich media. because we don't know the composition of that media there. And because serum actually consists, if you are extracting a serum from a particular organism or an individual of an organism, or from a different individual of the organism, the composition of serum might vary, because the serum composition will vary from individual to individual. And we only know that there are some growth factors or any cytokines that or hormones that are present within that particular serum that can induce cell growth and differentiation. But how much that of that particular component which is present within that serum we might not know, or if there is an additional component that is present within that serum and not from the serum obtained from the other individual, we might not know. So that's why this B27 is a defined media and not it does not consist of any serum, free serum there. So basically it is used to support the growth and survival of primary neurons there, including those from hippocampus, because hippocampal neurons is something that is used to study neurodegenerative disorders there, right? So in case of fetal growth and all, B27 is used there. So it consists of a defined mixture of vitamins, antioxidant, fatty acid that enhances neuron viability in culture, while G5 is used to support glial cell growth rather than neurons there. So basically, if you look into glial cells, the several different glial cells are there, right? What you called as microglia is there, oligodendrocyte is there, as well as astrocyte is there, astroglia or astrocyte is there. So basically, rather than neuron, G5 is to support that particular media. It is also a defined media there. So basically, if you look into that B27 media or NB media, neurobasal media, it is called as NB27. And 27 represents the amount of number of substances that is present there. There are 27 different substances that are present within that media. That is why it is called as B27 or NB27. So it consists of the sodium bicarbonate to maintain the pH there. Apart from that, amino acid, vitamins, etc., glucose, etc., to provide the energy source are also present there. This I have discussed before. NB or B27 is used to grow prenatal or fetal neuronal culture, while neurobasal A media is used to grow postnatal after birth and as well as adult neuronal cell. It consists of low osmolality, about 220 milliosmoles, while in neurobasal A media it consists of about high osmolality, like 260 milliosmoles, and it is used to grow embryonic cell, as well as in this case it is used to grow mature neurons. And specific supplements are there for optional growth, and also based on how to grow an adult neuron, commoners in NB27A also varies. So I suppose this is clear. So does anyone has any doubt, or should I explain it once more? So serum free, like fetal culture media, fetal neuron culture media. So I suppose this is free. I suppose this is fine. I think it's quite understandable. So I will just move on to the next question. Coming on to question number 10. Read the following statements carefully. What is an animal origin media? So this is the option: is not manufactured using any raw material and ingredients derived from animals, including human tissues, cells, or body fluid at the primary level of manufacturing. No animal derived materials are used as starting material or in primary level manufactured process. That is the option number 1. So second option is that second point is that, sorry, not option, it's a point. Second point is that the component used to manufacture the raw material ingredients are not derived from any animal tissue, cell, or body fluid at the second level of manufacturing. Ingredients and components may be produced by or extracted from plants, bacteria or yeast, or self-free system derived from them, or may be produced by organic synthesis. Next point, the final point is may contain recombinant animal or non-human animal proteins, non-animal proteins provided they are expressed in plant or microorganisms. So first option, only A is true. Second option, only B is true. Only C and D is true. All are true. And getting option number 4, all are true. A, B, C and D are true. Any other option? Yeah, so basically you are right. All of them are true. A, B, C and D are true. So basically if you look into animal origin free media, so basically you cannot have anything that is derived from animal source there. So the first option is actually right there. because it does not have any something that manufactured from animals, tissue, cell or body fluid, whatever it is necessary. So that particular point is true. Second option, second point, the component used to manufacture the raw materials are not derived from animal tissue, cell or body fluid. That is also true. So it is also, that particular point is also true. So both A and B are correct. Ingredients and components may be produced by or extracted from plants, bacteria, or yeast or cell-free system derived from them. So it is also actually called as animal-free media. Any other component that is not present in the animals can be introduced into it, whether it's from plants, bacteria, or yeast, or any cell-free system can be used in that media to make that particular media there. So basically that is also true. May contain recombinant animal or non-animal proteins provided they are expressed in plants or microorganisms. Recombinant animal sources can be used not direct from animal but indirectly from animals it can be used. So that particular option is also true. So the answer to this question is option A, B, C and D are true, or all of them are true. So does anyone have any doubt regarding this explanation? So I think it's quite clear. So I will just move on to the next question then. Coming on to question number 11. FACT stands for flow assisted chamber system, flow assisted cell sorting, floor for attached column separation or flow of arterial chronic segments. Option number 2 I am getting. So basically you are right. So FACTS we have discussed earlier that also, right? It is also called as fluorescent assisted cell sorting. So basically from a sample of cell or like a population, different population of cell, if the sample you are having consists of a different population or cell population there, so it is passed on to a small tube that is present within FACS there. That small tube only promote the flow of only one cell at each time point. So then it moves on to as it passes on through a particular tube, it is then irradiated by a laser that is provided from a source there, right? So after that it gets scattered, the laser gets scattered once it touches a particular cell, and based on the property of the cell, whether it is densely packed, what is the size of the particle is, what kind of receptors or what kind of fluorophore, if it is attached to any particular fluorophore or anything, then it actually gives out a scattered signal from the laser that is touching the cell surface there. So based on the scattering, obtained that is detected by the photomultiplier tube or any kind of detector system, this particular information is then transmitted to a computer there which analyzes the data. So basically a softwares are provided by different vendors that actually gives you this facts and all. So once this information is received, so it gives out an idea about the granularity, how much dense the cell is, what kind of cell population it might be and everything, which all diverse populations are present there. So it gives you an idea there. So that it not only that, once it passes out the tube, it then enters out at smaller droplets there. So this smaller droplets is getting ionized there. So once ionized, it is then moved on to a like electromagnetic field there. So when it get ionized, it will get a positive charge, negative charge, or it will be there will be no charge at all. So based on that, it is then separated into different sample vials there. It can move into a negatively charged electrode if it is positive charge. It can move on to a positively charged electrode if it is negative charge. The cells are negative charge. If it has no charge at all, it moves into a different tube there. So as it moves into different tube, it is then transported onto different vials that are end vials that are kept there, so that population can be separated out there. So that's the basic function of FACS there, or fluorescent assisted cell sorting there. So does anyone have any doubt regarding this? Okay, I think this is quite clear. We have discussed in the first question itself. So I will move on to the next question. Immunopanning is a technique used for cell attachment, cell death or senescence, cell size and number calculation, cell selection and separation. Which might be the answer? Okay, I am getting option number 4. We have discussed this before. Okay, I'm looking at in the option number 4, sorry. So you are right, basically it's the answer is selection as well as separation. So basically immunopanning we have already discussed. I will just recall it once again. So basically if you have a population as to different cell population, if a sample consists of different cell population, you are transferring that into a cell culture plate, or a cell plate which consists of certain antibodies attached to it. So before that we are actually adding, suppose that we want to separate leukocytes, so it consists of a CD45, CD42 marker, right? So if you are targeting this antibody that actually binds, if you have an antibody that actually targets this particular CD marker there, cluster of differentiation marker there, it actually binds to it, forming an antigen antibody with the cell, right? So after that, this only this leukocyte that is present within the cell population binds with this, interact with this antibody there. So this this sample entirely is transferred into a culture, or this particular plate there. So this plate is covered with at the surface with a secondary antibody there. So the property of this secondary antibody is that it has a tendency to detect the crystalline or the Fc region of this primary antibody there. So the cell is bound to this primary antibody. When it is transferred into this particular dish or the plate there, only the cells that is bound to the primary antibody will be bound to the secondary antibody. All the other cells will remain in the suspension there. Then afterwards we are removing some of the suspended solution that is present at the surface. Afterwards you are washing the remaining solution there, or washing the remaining dish there, and then using a particular detergent or any buffer there, mild washing is given there. If you are giving a harsh washing, then all the primary antibody that is interacting with the cells will be lost out there. Basically we don't want that. So you are giving a mild washing there. Then all the remaining contaminants or the unbound cells will get washed out. Only a bound cell with the antibody is getting present in the dish there. Then afterwards you are adding a elution solution there, so that the antibody interaction is getting broken there. Then you are collecting the suspension, so the antigen and the cell that is, or the antigen that is bound to the primary antibody is getting released there into the suspension. Then you are collecting the suspension and then transferring into a sample collection tube there. So this is what immunopanning is there, similar to density gradient cell centrifugation as well as FACS. It is also used to separate out a cell population, a particular cell population from a mixed number of cell populations there. So I hope it's clear. So should I explain it once more, or should I move on to next slide? Okay, so I think it's quite clear, so I will just move on to the next question. So which of these following is not expected from the dissection of a spinal cord of E14 or embryonic day 14 rat? Skeletal muscle myoblast, motor neurons, glia, glial cells, or DRG? It might be the option. Okay, I'm getting option number 4. Are you sure? Like any other option there? E14 basically. Okay, so the answer to the question is skeletal muscle myoblast. Basically these are all part of the nervous system, motor neurons, the glial cells, as well as DRG or dorsal root ganglion there. So basically you are basically dissecting these particular neurons either from the motor neurons in case of this peripheral nervous system or a spinal cord, and glial and DRG also in case of peripheral nervous system, and the glial cells in case of central nervous system, that is from the brain. So these are usually isolated at the E14 embryonic day 14 rat, embryo there. So basically it is the perfect time to harvest neurons from the rat cell or the rat embryo there, because afterwards the cells can, these neurons can undergo a programmed cell death or apoptosis. So at this stage, embryonic day 14, it is at a high purity. Afterwards, it starts to undergo apoptosis. So after that days, it then also starts to become a fetus there, right? So this is the preferred date, that the embryonic day 14, starting from embryonic day 0, that is the time of gestation, from the starting point of gestation period itself, or the carrying period of the mother. So basically, if you look into skeletal muscles are not myoblasts. These are basically used to culture muscle cells, right? So basically, they are obtained from hind limbs of a fetus or from an adult rat there, right, after the postnatal time and all. So basically, these are not present in the dissection of a spinal cord. These are present in the hind limb of that particular embryo or the fetus or the adult rat there. So and is not located within the spinal cord. All the others are located in the spinal cord. Motor neurons should be dissected from this A14, as I said, because afterwards it is the time point where they are present at the high purity, and afterwards they start to undergo autophagy. Autophagy or apoptosis or programmed cell death there. So afterwards you cannot do that. At this particular time point, you have to isolate them out. So dorsal root ganglion, if you see that, is basically a collection of neurons. Basically, it is sensory in nature and is present at the dorsal root of the spinal nerve. So basically, I have shown you the image. I will show you that again. Basically, so basically, this is the image there. So basically, this is the spinal cord there, right? So you can show that this is the dorsal root. So this consists of this cluster of this neurons there, and it's basically similar to that of what you say motor neuron there. It is sensory in nature. I will just repeat this out. So, and it is located at the spinal nerve. It is part of a peripheral nervous system itself. So, and it is sensory in nature, as it was said, and it consists of pseudo-unipolar neurons there. So basically, if you look into different types of neurons, it can be unipolar, bipolar, as well as multipolar in nature. And one other term is pseudo-unipolar. So if you look into unipolar neuron, it consists of a single axon that is arising from a single cell body. If it consists of two different axons arising from two different points of a cell body there, then it is called as a bipolar neuron there. If it consists of different axons that are arising from different points, then it is called as a multipolar neurons there. So what is a pseudo-unipolar neuron? Meaning is that it acts as a unipolar neuron there, meaning that a single axon develops from a cell body there, single cell body there, then the axon branches out into another branch. So it consists of a single axon arising from that, then like a tree trunk, it branches out. So that is why it's called a pseudo-unipolar unibody, because it turns out to be a unipolar, but it is not unipolar because it branches out into two different branches there. So as I said, it is sensory in nature. So it is a part of a nervous system there. It should be started at T14. So if you compare the glial cells there, you can see that glial is one another remaining. So basically, astroglia and astrocytes are one type. Astroglia is there, microglia is there, oligodendrocytes is there. So astroglia is a component of central nervous system. These all are components of central nervous system, these glial cells. So basically, astrocytes play a major role in regulating the homeostasis of the central nervous system. Like it helps to regulate the ion balance, remove extra neurotransmitters like glutamate, and also provide metabolic support to the neurons. Microglia actually acts as the primary immune defense. So it is also called as a brain's macrophage there, because it can act as macrophage. It can have the capability to phagocyte like foreign antigen molecules there. It has the capability to undergo phagocytosis there. So it is actually a defense cell there. It is required for immune defense there. So they monitor the CNS for pathogens and debris, respond to inflammation, and also other protective and damaging roles, prevents the protective and damaging roles there. And oligodendrocyte, if you look, like a Schwann cell that is present in the peripheral nervous system that helps in the formation of a myelin sheath there. Oligodendrocytes are actually present within the central nervous system. Those Schwann cells are actually present in the peripheral nervous system, not in the central nervous system. These oligodendrocytes are actually present within the central nervous system there. So, and instead of Schwann cells there, oligodendrocytes are the one in the central nervous system that actually helps in the formation of myelin sheath to form myelinated neurons within the central nervous systems there. So these are the glial cells that perform these functions. So does anyone has any doubt, or should I explain this once more? This we have discussed earlier also in the last or previous week. So I think it's quite clear. So I will just move on to the next question then. Coming on to question number 14. Morphology of cell tells you about number of cell, the shape or form of a cell, number of doubling by cell population, or a biochemical constitution of the cell, which might be answer. Yeah, I'm getting option number 2. Yeah, basically morphology you all know. Basically you are right. It tells you about the shape or form of the cell. It cannot tell you about the number of cells there. Basically you are looking into a single cells there or a population of cells there, then you are dictating a morphology there. From that morphology you can also try to distinguish whether it is a nervous cell, whether it is skeletal muscle there, whether it is a heart muscle, whatever it is, based on the morphology. So basically it does not give an idea of number of cell, it gives an idea of shape or form of the cell. Number of doubling by a cell population you cannot study there. Basically it is also called as a generation time, right? How many, like doubling time or generation time. It is a totally a different parameter there that you can study from a growth curve there. If you are studying the number of cells there, you can basically point out the growth curve. You can study the number of cells that is formed at the time range and then plot a graph there. From that you can actually calculate the doubling time there. So morphology doesn't give an idea of number of doubling by cell population. Biochemical constitution, actually you are going into the molecular biology of the cell or a biochemical aspect of the cell. So from the morphology of a cell that you are getting, you cannot basically detect that, or you cannot basically study that. So the answer to this question is morphology of the cell tell you about the shape or form of the cell. So does anyone have any doubt, or should I move on to next question? So I think it's quite clear. So I will just move on to the next question then. Coming on to question number 15. Insulin is secreted by liver, pancreas, bilirubin, or gallbladder. Which might be the answer? Yeah, I'm getting option number 2, pancreas. Yes, basically you are right. The answer is pancreas. Basically it's a basic question there, right? So you might have the answer. So apart from pancreas, apart from insulin, pancreas also secrete an hormone called glucagon. Both of these are hormones, including insulin, which is a hormone. So basically it is used to regulate the blood glucose level that is present within the the glucose level that is present within the blood, right? So basically insulin, what it does is that it tells the cell to uptake the sugar that is present within the blood, right? If there is high level of sugar within the blood, then the cells can actually utilize this glucose that is found within the blood instead of glucagon, which does the opposite. It tells the cells to break down the glucagon that is present in the body, and then other components that are present in the body, and then convert into glucose, right? So these are two antagonistic hormones there. One acts in reducing blood glucose level, other acts in increasing blood glucose level there, right? So, and these are hormones basically. Both of them are hormones that are required for our maintaining the physiological activity of our body there, and is secreted by this particular organ, that is pancreas. So yeah, so I hope the answer is clear. So should I move on to the next question? Does anyone has any doubt? Okay, so I think the answer is clear. So answer to your question is insulin is secreted by the organ pancreas. So coming on to question number 16. Which of the following only have an electrical activity but not a mechanical activity? Neuron, cardiomyocyte, skeletal muscle myotube, or all of the above? only electrical activity but no mechanical activity. Okay, I'm getting the option number 1, right? So basically, right, the option number 1 is neurons. Basically, it shows the electrical activity within there, right? So because electrical conductance is something that helps the neurons to communicate with each other. So basically, if you are comparing electrical activity, it is not only there, chemical activity is also there, because within the synapse there, right? So synapse is something that is present within a junction of the nerve endings of one neuron to the dendrites of the other neurons, right? So in order to transmit signal from one neuron to the other, it has to pass the synapse and then move on to other neurons there, right? So basically, in that case, in the neurons, instead of electrical activity, there are something in the synapse. Instead of electrical activity, there is something called as neurotransmitters that are present there, right? Acetylcholine is there, glutamate is there. So what this does is that it acts as a chemical messenger from starting from, say, the nerve ending of first neuron, then it moves on to the dendrite of other neuron, then transmit the signal there, right? So based on the presence of these neurotransmitters there, the relay of signaling from one neuron to other take place within the synapse there. So basically it's an electrochemical activity, not just an electrical activity. And in case of cardiomyocyte and skeletal muscle myotube, you can see that cardiomyocyte actually have an electrical activity there, right? So in our heart there is something called as Purkinje fibers there, right, which is called as pacemaker of the heart. This starts an electrical signal there. So this spreads to other cardiac myocyte and this continues on, and then the heart starts to show some mechanical activity, starts to beat. It starts to beat in and out. So this is what the electrical activity actually maintains along the mechanical activity of the heart muscles there. So in case of skeletal muscles, there also similar activity is there. The electrical activity is there, and then it actually has to carry out the mechanical activities there. Sliding filament theory is something that happens there also. And so the answer to this question is which of the chemical only have an electrical activity but not mechanical activity in this case there, right? So the answer to this question is neurons. So I think I will just move on to the next question then. So coming on to question number 17, which of the following is the smallest functional unit of skeletal muscles? Skeletal muscle myoblast, satellite cell, skeletal muscle myotubes, or skeletal muscle myonuclei? Which might be the answer? Okay, I'm getting option number 3 and 1. 2 also I'm getting. Okay. You have to remember the question, smallest functional unit of skeletal muscle. Functional unit, smallest. So that gives you a doubt there basically, which might be the option. So the answer to this question is skeletal muscles myotube, that is option number 3. Okay, so yeah, option number 3. So basically, if you look into the first option, that is a myoblast, these are mononucleated precursor cells that can proliferate and differentiate into myotubes. So you can see that these are the myoblasts, right? Satellite cells are the progenitor cell. It acts as a stem cell to generate out other skeletal muscle precursor there or myotubes there. So which in turn forms a muscle fiber there. So this satellite cells actually is a progenitor cell. It doesn't carry out the functional aspect of a skeletal muscles there. So basically it actually forms the skeletal muscles there, but if you look into the functional aspect of a skeletal muscle, it is not carried out by the satellite cells there. It just forms that skeletal muscles there. Myoblast is also something there, because myoblast is something when this satellite cell forms, right? So it starts to grow and differentiate, then it starts to aggregate together to form a group. Then it starts to lose its identity and then they join together to form. So this particular segment is called as a syncytium or a myoblast there. They group together into a particular structure there. This is called as a myoblast there. So it is actually a precursor or a product that is formed from the proliferation activity of satellite cells there, proliferation and grouping activity of satellite cells there. So it is not the functional part of that particular skeletal muscles there, because it actually is a particular aggregation of cells that is formed there. Coming on to skeletal muscle myotubes, these are formed by the fusion of multiple myoblasts and enucleated basically there, and this aggregates to form what is called as a muscle fiber there. So these have the tendency to what you call contract and relax. It will have that particular myosin and actin filament fibers that are present, that should be present within a muscle fiber there, and then these myotubes actually combine or group together to form what is called as a muscle fiber or a skeletal muscle there. So basically this is the functional unit, smallest functional unit, because if you look into microscopic view of each and every myotube, it has that particular myosin, dark band, light band there, that are aggregated within a particular form, and it can have the ability to contract and relax there. So it carries out that particular function, the function to actually contract and relax according to the stimuli that is received there. So that is the smallest functional unit. The myoblast and satellite cells are not, because they have the ability to divide or form muscle cells, or the smallest functional unit of muscle cell, but the smallest functional unit of a muscle cell is the myotubes there, rather than the first two options there. And if you look into a myonuclei, these are basically nuclei that are present within the myotubes or a muscle fiber there. So that's a different kind of option, so that's not the answer. So does anyone has any doubt there? Like in case of central nervous system, neurons are the smallest functional unit, right? Case of kidney, nephrons are the functional unit, right? Smallest functional unit. So in case of muscles, myotubes are the smallest functional unit there. So I think this is quite clear. So should I explain it once more, or anyone has any doubt? So I think I will move on to the next question. I think this is quite clear. You have to just read the question carefully, like that's the important thing. Coming on to question number 18, you will know the answer to this question. Which of these are skeletal muscle stem cells? Skeletal muscle myoblast, satellite cell, skeletal muscle myotubes, or skeletal muscle myonuclei? Which may be the answer? Option number 2, I am getting any other option? Option number 2 I am getting also. So majority of you are answered the satellite cells, and you are basically right. So I think I should not explain this once more. Basically it is, explanation is quite clear, because I will just explain the satellite cells, because these are stem cells that are present within the muscle fibers area. So if any injury or any repair happen, what you say, like these stem cells become active, then it start to divide and regenerate to form what is called as a myoblast there. So these are actually like this stem cell like activity is carried out by satellite cells there, and these are what we look for when you are doing a cell culture incubation. And if you are doing a a muscle fiber there or a skeletal muscle within a culture plate, you need to isolate these satellite cells, and then you are putting a nutrient media, and then you are incubating it out. So that's why, because it is a progenitor stem cell there that can give rise to skeletal muscle cells. Sometimes you also add fibroblasts, because fibroblasts also secrete some of the growth factors that are required to make these skeletal muscle cells there. So fibroblasts actually divide faster, so like the number of fibroblasts that are introducing into a culture plate should also be maintained there. It should not be much larger, as it might outnumber the amount of skeletal muscles that is formed. So basically these are isolated at the postnatal stage or at the fetal stage from the hind leg of a rat baby there, right? So yeah, so this is the answer basically. Which of the following are not skeletal muscle stem cell? The answer is satellite cells. So does anyone have any doubt, or should I move on to the next question? Okay, I think the answer is clear. So I will move on to question number 19. Which of the following is multinucleated? Skeletal muscle myoblast, satellite cell, skeletal muscle myotube, skeletal muscle myonucleus. Which might be the answer? I am getting the answer option number 3. Yeah, so basically the answer to this question is skeletal muscle myotubes. You are right basically. So basically, if you look into the structure there, right, you can see that this is the myotubes, right? So basically, if you look into myoblast and satellite cells, they are uninucleated. There is only one nucleus that is present there. But in case of myoblast, when the myoblast actually aggregate to form what is called as a myotubes there, right? So that is a single function, that is acting as single functional fiber unit there. So if you are looking at that as a particular unit there, then it consists of several nuclei that are present within that particular myotube itself there, right? So basically that is multinucleated in nature. So these are formed by the fusion of multiple myoblasts as it discussed here, and are therefore are multinucleated. Multinucleated there. So basically the answer to this question is, which of the following is multinucleated? The answer is skeletal muscle myotube. So does anyone has any doubt regarding this? So basically this multinucleated means the nucleus that you are referring to is myonucleus, skeletal muscle myonucleus. So this each nucleus is called as skeletal muscle myonuclei. So I suppose that this is clear. So I hope there is no doubt. So I will just move on to the last question. Cell digestion in cell culture is done using trypsin, which is produced by the human body, is produced by which organ? Liver, pancreas, bilirubin, or gallbladder? Which might be the answer? This is the last question. Trypsin is produced by. Okay. So the answer to that question is pancreas, right? Along with the hormones like what you say, insulin and glucagon, these also produce certain enzymes, trypsin, which is actually proteolytic in nature. It's a serine protease, right? So it actually have catalytic triad made up of this serine, aspartate as well as histidine, I suppose. So this actually cleaves the basic amino acid terminal or carboxyl region of basic amino acids like lysine or arginine, right? So whenever it cleaves at this region, the proteins that are already present there is actually forming peptides. So it is degraded down into peptides there. So basically it is used as this particular enzyme, which is proteolytic enzyme, is used to lift off cell culture or used to separate out a tissue into individual cell, which can later be introduced into a culture plate to carry out the cell induction and everything. And when you are doing passaging and all, when you are used to remove the cells that are present within a culture plate or degrade out the extracellular matrix, you are adding trypsin, collagen or collagenase, papain, etc. And if you are using trypsin, which is much more active in nature, after some time point you need to add trypsin inhibitor to inactivate it. So in the human body, it is used for carrying out the digestion process and everything, so that our body can uptake the amino acids that are present within the food material and all, right? So cell digestion in cell culture is done using trypsin, which is the human body, which in the human body is produced by the organ called as pancreas there. So yeah, so I think this is quite clear. So does anyone have any doubt regarding this? Or from the overall topic, do you have any doubt that needs to be cleared out? So yeah, I think it's quite clear. So I will just wind up the section for today. So thank you all for attending the class for today, and I think it's quite clear. There is no doubt. Okay, so I think you can meet for the next week. One more section will be there, I suppose, if possible. So try to join that also, and if you have any doubt, you can ask at that time point also. So yeah, thank you.